Hello Year 12s, welcome back. It's Mr. D. We're going to continue on with a recap of our last video. So we looked at the three stages of gene expression. We've covered transcription now. We've covered RNA processing, and we're going to finish with translation today. So what is translation? It's essentially the creation of the polypeptide chain, which is the protein, through the decoding of the mRNA strand via a ribosome. Cool. We looked at RNA processing. That's where we have essentially our RNA strand that's been coded from our DNA. All right. And the introns are going to be removed because the exons are expressed. All right. And of course, we have the last part here, which is the 5' methyl G cap added to the 5' end and the 3' poly A tail added to the 3' end of the messenger RNA. So again, we're covering gene expression. We're just going to look at translation today. And these are our dot points as per usual. So what is translation? So as you can see, it involves this specific protein here, which is this odd T-shaped protein with an anticodon. What was that called again? You should remember that's called a tRNA, hence the shape. All right, and essentially these bad boys, they bring the amino acids that you can see at the top here to the ribosome, and it starts to build the polypeptide chain. So you can see that here. All right. That's one tRNA molecule with its anticodon. So UCA is obviously complementary to the AGU that you see there. Okay. So we have it here. tRNA, UCA, complementary to AGU. And that would have brought this purple, actually in this case the green amino acid here. Okay. The next one is going to be GCA. You can see it here, tRNA, and it has that pinky colored amino acid. All right, and obviously you have the bonds in between, which are your peptide bonds as well. So we have our overall process here. As you can see, we have our tRNA molecules, the ribosome, and the messenger RNA. Okay, and then you have the specific amino acids. So in this case, leucine, arginine. You can see they're a different shape. Okay, but part of them are not a different shape. Boom, boom. We know that that's the basic setup. And then the part in orange, okay, that's your R or side chain. All right, so as we can see here, we have our basic groups. So we have our nitrogenous group, our middle carbon, and then our amine group, as well as the side chain or the variable group, and that changes. What's circled here is the parts that will connect to one another, and that is how it creates the peptide bond. So remembering this is our ribosome, all right, it's just two different segments. Okay, and the messenger RNA will bind to that ribosome and will be fed through. Okay, and every codon, so every 3 bases, it's going to be red, and a tRNA molecule is going to bring the amino acids. They get chained together, and a chain of amino acids is just a polypeptide, and there's your protein. It's now been made. So something to note is that, remembering for when we looked at our codon chart, that every codon codes for a specific amino acid. Okay. All right. This is the same thing. Peptide bond shown here. All right. And that's made via a condensation reaction. That's not as important. The important part is to understand that it's a peptide bond. All right, because it's essentially what keeps the polypeptide together. So the language is the same there. So we have our codon chart here, our tRNA molecule with its specific anticodon. So what I want you to do now is figure out what the anticodon would be, and then figure out what the amino acid would be using the chart. Okay, I'll give you 10 seconds for this. If you said GAC, you were correct. which then means which one is going to be the one that's shown on the codon chart. That's right, cysteine. Remember, that was because it was a codon chart, not an anti-codon chart, which is the one found on the tRNA. Okay, this next image shows everything, the whole kit and caboodle, all being done within the cell. All right, so you have the cytoplasm, so this is your cell. You have this purple part here, that's going to be your nucleus. All right, and then you have your boxes showing each step. So number one, transcription occurs over here. All right, within the nucleus, and then it goes out of the nucleus. So within the nucleus, all right, the chromosomes get uncoiled, the DNA gets split, transcription occurs, and our messenger RNA is then created. That then leaves through the nuclear pores as pre-messenger RNA. Okay. RNA processing then occurs where it will get a 5' methyl G cap added to one end and a 3' poly A tail added to the next, and of course the introns get removed. Okay. Then lastly we have translation, okay, which we just looked at this whole entire class. So that was the mature messenger RNA that now has been changed through RNA processing, then gets attached to a specific ribosome that could be free floating in the cell, or it could be on the rough endoplasmic reticulum, for those who know the organelles. And that will bring your amino acid across. The tRNA will. Okay, those get joined together with a peptide bond to make our polypeptide chain, which is then our protein. Beautiful. And the last thing to note is that there is actually post-translational modifications that occur to these polypeptides. So once they're made in the ribosomes, they're going to get transported through a vesicle, all right, somewhere within the cell. And in this case, it's the Golgi apparatus. Sometimes they might get transported outside of the cell through exocytosis, or they might go through a vesicle, transport vesicle, to the Golgi apparatus. Okay, and here there's a few different things that can happen to it. Okay, again, this is more chemistry based, but it's still part of biology, so you should be aware of it at least. You should be definitely aware that there's modifications that occur. Okay, so what can happen? Methylated, so adding a carbon and hydrogen, well, carbon to 3 hydrogens. Acetylated, carbon, 3 hydrogens, carbon and oxygen. Phosphorylated, phosphate added. Glycosylated, so some sort of sugar is added. All right. So some sort of saccharide or polysaccharide. Or lipidated, so a lipid can be added. Some sort of fat, so it could be free fatty acid or glycerol there. All right, so the most important part of this slide is just to understand that the Golgi apparatus then modifies the protein, and there's a few different things that can happen. Okay, that's one of the special roles of the Golgi apparatus. When they say in year 11, like packaging and transport, this is your packaging. Okay, alrighty, thanks for listening. Have a nice day. The next one's going to be big.